Okay, live stream is ready to go.
Okay, so I think we can start now.
Sorry.
There we go. I think we can start now. There we go. So welcome to the AFO Cafés. These are informal conversations about all things birds. And I am Valentina Ferretti, president of Association of Field Ornithologists. And because we usually have people that are not members of AFO, I'll just provide you with a brief summary of what we do. AFO was founded in 1922 in Massachusetts as a member-based organization dedicated to the study and conservation of birds and their natural habitats. We started out actually as the New England Bird Banding Association, then were later renamed the Northeastern Bird Banding Association, and then later AFO. And as our early names suggest, we have a strong interest in the study of banded bird populations. We also have a strong focus in Latin America through outreach programs, the organization of joint conferences in the neotropics, and grants. Next year, we will be celebrating our first 100 years. And we are organizing a meeting in Plymouth, Massachusetts in October 10 to 13. So save the date. And if you're a student or early professional, we are going to have grants, travel grants and registration grants for the meeting. So make sure you check our website. At AFO, we actually offer cafes. These are monthly events, and our next cafe is going to be on November 19th, and it will be given by Matthew Gonerman on wild turkey land use and at their northern range limit. Our cafes are sponsored by AVNet Research Supplies, and this is our actually a store that it's related to AFO, and we sell research equipment, field equipment, mist nets. So you should check our website that's on the page, on the screen right there. It's avnet.com. If you want to, if you're looking for some field equipment and research equipment. Today's cafe will be presented by Nicolas Lois and Dr. Rebecka Brasso. They will talk about stable isotopes and mercury concentration as ecological niche proxies in the Southern Rockhopper Penguin. Dr. Nicolas Lois, very recently, actually less than a month ago, defended his PhD and from the University of Buenos Aires on this very same topic. And for his PhD, he had a CONICET fellowship in Argentina. CONICET is our national research council. I think that's a fair translation. And conducted his work at the IEGEBA, that's the Ecology, Genetics, and Evolution Institute of Buenos Aires. He also has a teaching position at the Department of Ecology, Genetics, and Evolution of the Universidad of Buenos Aires. And Dr. Rebecka Brasso is an assistant professor at Weber State University, where she studies heavy metal contamination across terrestrial, aquatic, and marine food webs. Dr. Brasso received her master's at the College of William and Mary and her PhD at the University of North Carolina at Wilmington. Now, if you enjoy our AFO cafes and programs like this, become a member. You can look on our website, and your membership actually supports programs like this and also the organization of meetings and grants and other very interesting things. And with that, I will just leave, I'll stop sharing my screen and I will leave it up to Nicolas, I think. He's going to start.
I will start us.
Oh, okay. Sorry.
It's okay. Oh no. Am I sharing my screen? Can we see it?
No.
No, not yet.
Okay. How about now?
Yes.
Okay. Right. Sorry, you would think I would know how to do this at this point, but here we are. So thanks so much for having us today. And I'm really excited to talk about what I think is a really cool science story, in that for me, this research stems back to my dissertation, which I finished back in 2014. And so what we're going to do is I'm going to tell sort of the first half of the story of how this project came to be, and introduce some of the background. And then Nico will pick up sort of where I left off. And so I think that that's one of the things that I really love about this project, is that I think we all always wish that we could have answered all the questions that we wanted to and sort of kept going and going. But in this case, I've been really fortunate to have wonderful collaborators who've sort of kept this work going beyond my project itself. And so the focus of the research that I started for my dissertation was looking at mercury contamination, and specifically looking at it in marine ecosystems, and then even more specifically looking at polar marine ecosystems. So just to give a background on mercury itself, so mercury is a naturally occurring heavy metal, and that there are a number of natural sources. So things like volcanic eruptions, degrading limestone, wildfires can add it back into the global cycle, geothermal activity can all add mercury into and sort of kick it back up into that global cycle. But today, more than half of all new mercury emissions into the atmosphere come from anthropogenic or human sources. Primarily, we're looking at things like coal burning, but these days, increasingly, we see a lot of new emissions coming from artisanal and small scale gold mining activities. So once this mercury is added into the atmospheric cycle, it exists as a gas, and as a gas, it is subject to long-range transport. It can be transported globally because it has a really long atmospheric residence time. So estimates have put it atmospheric residence of up to over a year for gaseous forms. But since we're more interested in what's happening in ecosystems on land, essentially what happens is that this mercury as a gas is transformed into an inorganic form in the atmosphere. And then that form is deposited either by wet or dry processes into terrestrial or aquatic systems. And particularly aquatic and marine systems are of interest because it is in these habitats that mercury is readily converted from its inorganic form into the more toxic and bioavailable form of methyl mercury. So in these aquatic and marine systems, there are large communities of methylating bacteria, primarily things like sulfate-reducing bacteria and iron-reducing bacteria, that are going to take this inorganic form of mercury and convert it into organic mercury or methyl mercury. So methyl mercury then is able to be incorporated into the food web. And not only is it incorporated into the food web, but it goes through this process called biomagnification. And biomagnification is where we see an increasing concentration of mercury with each step in the trophic ladder. And so this makes the organic form of methyl mercury much more toxic to organisms than the inorganic form. Inorganic mercury can accumulate within an individual, but doesn't often biomagnify through food webs, whereas the organic form of methyl mercury does. And so if we look at my little model here of a sort of traditional marine food web in the polar region, okay, if you will. So here I have methyl mercury as the little plus sign. Once mercury is deposited into these systems, the bacterial communities are going to rapidly convert it into that organic or methyl form. Once that occurs, it's going to be picked up by our primary producers, or our plankton, that are grazing on that bacteria. And by the process of biomagnification, there's going to be more mercury in those primary producers than there were in the trophic level below them, those bacteria. So then we can go on up to our primary consumers. So a lot of times our crustaceans, things like krill, okay, more biomagnification, higher concentrations in our fish. Okay, and then ultimately up into our apex predators, which in these marine systems that we're going to be talking about are the penguins, okay? And then ultimately big top apex predators like seals or toothed whales. And so again, by this process of biomagnification, mercury concentrations are lower at lower parts of, or at lower levels of the food web or low trophic levels. And then we get this exponential increase in concentration at higher levels. This is why it's typically cause for concern. Higher concentrations. Why it's typically cause for concern for predators having higher concentrations, as higher concentrations of mercury are more likely to have harmful effects on things like reproduction and basic physiology. So one of the things that's been a really helpful part of the toolkit in understanding not just mercury concentrations, but the how and the why of why certain species have different concentrations of mercury and how that occurs is using stable isotopes. And in particular, Nico and I both use stable isotopes of nitrogen and carbon in order to quantify trophic level and foraging habitat. And so the first one that we're going to be talking about today are stable isotopes of nitrogen, and we'll refer to it in this delta N15, sort of in this delta N15 version. The way that nitrogen works and that delta N15 works is that we expect to see an increase in the proportion of the heavy isotope of nitrogen, nitrogen 15, to the lighter isotope of nitrogen, nitrogen 14, as we go up the trophic ladder. So within any given food web, organisms that are at the base of the food web will have lower delta N15 values, and those that are higher up in the food web towards our top predators will have higher delta N15 values. And so within a food web, this allows us to quantify trophic level or relative trophic position between different organisms. Since mercury biomagnifies with trophic level, and delta N15 also increases with trophic level, we can look at the relationship between these two sort of as giving us a correlation or even a causality, okay, of why mercury concentrations might be higher in one species or population than another. So for example, our sort of predicted relationships are seen in this graph on the bottom left-hand side of the screen, where we would expect mercury concentrations to increase with increasing trophic level. And so again, if we find differences in mercury between species, we can use trophic level to understand, or trophic position, to understand or explain why those differences may occur. And again, based on that diet and the process of biomagnification. But it's not just what you eat. And more we're finding this. And in particular, it's very interesting in the marine habitat because it is three-dimensional and so dynamic. So in terms of mercury exposure, it's not just your trophic level, but we can also look at where you're foraging. And actually where you're foraging can play a really significant role in your exposure to mercury. The rate and degree to which inorganic mercury is converted into organic mercury depends on a wide variety of environmental factors: light, temperature, pH, dissolved organic matter. All of these qualities are going to vary widely across different environments. So for example, do you feed coastally or near shore, like this very lazy Gentoo penguin, which I can only assume is looking for krill? Don't bust my bubble if that's not what it's doing. I don't want to know. When I watched it, that's what it was doing. Or are you foraging out in open waters? Okay. Are you epipelagic, foraging in the surface waters of the oceans? Or are you foraging mesopelagic or on mesopelagic prey that's slightly deeper? Do you forage in an enclosed bay system, which may be more subject to having more organic matter as it washes off of that terrestrial area, leading to differences in mercury availability? Or do you feed out in the open ocean away from any coastal environmental processes that may alter the conversion or bioavailability of methyl mercury? So since mercury can also be differentially available based on environmental conditions, it sets the stage that we should check into and monitor where these animals are feeding in order to understand mercury variation. To do this, we use stable isotopes of carbon. And so here we're going to designate this as delta C13 as using these isotopic values in order to understand foraging habitat at sort of two scales. The first scale you can see here in this figure, in which we can look at a global scale of patterns of delta C13, in which we see less negative values in the tropical regions and more negative values in the polar regions. Okay, so where we're going to be talking about today, mostly in this region down here of South America. So again, we can look at broad scales, okay, of geographically where organisms are feeding. But for this project, and what was most interesting to us, was using delta C13 to indicate smaller scale processes of foraging. In particular, we were interested in looking at inshore versus offshore foraging, as well as benthic pelagic. So were you feeding on the bottom, okay, or closer to the surface? And so the figure that I have over here, I pulled from another paper that in which all I wanted to show here is that you can see along the x-axis less negative carbon values were associated with inshore foraging, more negative values were associated with offshore foraging in this variety of penguin species. And so if we look at what we would hypothesize for this research, right, and we put together our trophic tracer of delta N15 and our foraging habitat tracer of delta C13, we could look at trophic position, which is expected to show biomagnification of mercury from low to high. But then we could also look at whether mercury is related to foraging offshore or inshore to see if that was playing a role in the differences in mercury concentrations we were seeing. And so this brings me to this part of my dissertation in which I actually sort of put together all of these different tracers. And one of the big things that I was interested in doing was using penguins as biomonitors of mercury of food webs throughout the Southern Hemisphere. In blue in this map shows sort of a gross estimate of all of the different locations in which you can find penguin species breeding throughout the Southern Hemisphere. And so these are very phylogenetically similar and closely related species that forage in very similar types of habitats. And so I was interested in looking at the disparities in mercury among these populations. So for my dissertation, most of my work focused on the Pygoscelis penguins in the Antarctic Peninsula, but through collaborations, in particular with Dr. Andrea Rey at Cadic in Ushuaia in southern Argentina, I was also able to gain access to populations of Magellanic penguins, samples from Southern Rockhoppers. I did some collecting in South Georgia, and then through collaborations with other researchers at Phillip Island, I got little blue penguin samples, as well as African penguin samples from South Africa. And what I did was I ran all these feathers from the adult birds to look at mercury concentrations. And so to jump right to what I found, because that sets the stage for Nico, so in this figure, I have the different species across the x-axis and the adult feather mercury concentration on the y-axis. And the colors of the bar represent the different regions that you can see in the map figure here. And so we can see some general trends right away when we looked at these mercury concentrations in which our Antarctic species had relatively low mercury concentrations. But of particular interest to this project was a big surprise that we got here with our Magellanic and our Rockhopper penguins. And in fact, the Rockhopper penguins were extra surprising as they were the only population of all of these different groups that were studied that had mercury concentrations in their feathers that started to approach the lowest adverse effects level that's been documented for birds. And so this was certainly cause for concern, and we wanted to look a little bit more deeply into what was driving such elevated mercury concentrations in this population. And so for my work, I use trophic tracers in terms of using delta N15, and I use delta N15 to calculate a trophic level. And so in this figure, I have trophic level, okay, again, and this was informed by using the stable isotopes of nitrogen values. So low trophic level, up to high at 6. And then on the y-axis, the adult feather mercury concentrations. And these 3 populations of penguins, again, were all collected off of the coast of southern Argentina. 2 species at Staten Island are Magellanic, represented by SI, okay, and the Rockhopper. And then we also had Magellanic penguins from Isla Martillo, okay, that was closer inshore. But the interesting thing about this is that I expected the complete opposite trend to occur in this graph, because I know that mercury should biomagnify with increasing trophic level. But instead, what we found was that these rockhopper penguins that had the highest mercury concentrations of all populations that I studied actually were foraging at almost a full trophic level below the Magellanic penguins that were located very, very close to them. And so trophic level in this case did not explain any differences in mercury concentrations among these populations. And so that's where I kind of had to end my dissertation. And I kind of had to end it like this and just say, well, there's this uncoupling that's occurring here. And this goes against everything in my brain that says that mercury should align with trophic level. And so we ended with this, again, this uncoupling in which we had a low trophic level species that had high mercury. And so this sent us down this pathway and rabbit hole of wondering, could this be environmental? Was it the fact that there was something unique in the water, about the island, about the food web surrounding Staten Island that led to a higher environmental bioavailability of mercury that was completely unrelated to the trophic position of these penguins? So then I graduated, and then Nico came along. And so now I'm going to pass it on to him to give you the answer to this story.
Okay, can you hear me? Yep. So thank you, Rebecca, for the presentation. Just amazing. I hope I have an answer for this question. If not, we can keep talking and working. So Rebecca found this hotspot of mercury in the southern tip of South America, and so I continued her work and focused on rockhopper. But I want to first try to give some context of this species so that we try and understand all the factors that are involved in this mercury study that we did. So rockhoppers are a group of crested penguins. They are within the penguin family, and they have recently diverged, like just 2 million years ago. And we are focusing on this group, and mainly this group has been classified by IUCN as 2 species, and these 2 species are the northern, the one you can see in green here in the map, and you have these 2 populations in the Atlantic and the Indian Ocean, and then the southern, that is considered the other species is the southern, that brings together the southern and the eastern, so the ones you have in yellow and also the ones you have in this violet color, that it's they are vulnerable. So we have 2 groups, one that is endangered, one that is vulnerable, and that's how we started this project. So these birds, the species, possess an annual life cycle, which is characterized by 2 phases. So first they have this winter dispersal, when they live in the open ocean and they behave more like fish than like birds. And then they have this stage which is the reproductive stage, when they commute between the land and the ocean. They do foraging trips that last around from 1 to 15 days top. So they live here in this commuting kind of life, and it's gonna be important. Another thing I want to mention is that this is the molting period. So when we, as Rebecca told you, we work with feathers, so we are mainly focusing on this period of the annual cycle, but still the feathers are synthesized during this time, during the early molting, and they integrate all this time. And as regards mercury, the mercury is the main way of excreting mercury is through the feathers. So all the mercury that they could be acquiring for the environment, they will be mainly releasing it during the molt period. So that's an important aspect of this study. In general, this what we found, and we have recently published this paper with Coté Frugone from Chile. We found a global genetic structure that is separating this group into 3 species. The one that's in green is the northern, the same that is considered endangered by the IUCN, and we have found that these 2 groups should be considered species because they are highly diverged. And the 2 main hypotheses for this divergence is the subantarctic front working as a splitter of water masses in the Southern Ocean. So the ones that live in the south have certain characteristics, they prey on certain prey, and then the ones in the north have different water masses, template water. So yeah, and there's also the possible explanation of that during the glacial period of the last 2 million years, during these cycles, there could have been some isolation, and some glacial refuges could have existed in these places. So that's the other main explanation for this global structure. Within the species, we also found divergence, which is really interesting, and it will be really important in this work because we found huge divergence between these 2 populations, which is kind of logical because of the distance between these 2 areas, and they have no overlap between their foraging areas in the northern. So in the southern, we have also a lack of connectivity between these 2 populations, but at a really low, a really fine scale. So these colonies are separated by less than 700 kilometers, and we have found here a segregation. And we'll see now about this. We found, we knew that they have these non-overlapping foraging areas between these 2 populations, and there's also a hypothesis that many other species share, that is the potential isolation of Malvinas and the southern tip of South America during the glacial cycles during the last 1 million years. And another story happens with the Eastern Rockhopper, which has a higher connectivity and a higher degree of foraging overlap. And also there's a hypothesis that explains this high connectivity between all these populations and the lack of genetic structure because of a recent expansion. So they have been isolated longer during the last cycles, and they have not yet completely sorted out the populations, even though they are quite far apart. They could have colonized recently these colonies, these different colonies. So we focused on this southern on the southern tip of South America with the Southern Rockhopper, with this species that presented also a structure within a genetic structure within itself. And what we did was a thorough sampling of this area. We first sampled Isla Pingüino. That's a colony that has been recently founded. We also took the opportunity to sample some stranded individuals to try and assign them to the populations. And we found stranded individuals in 2016 from Tierra del Fuego and from Santa Cruz province here. And we sampled 3 colonies from Islas Malvinas, and we sampled in the southern tip, Isla de los Estados, the same sampling sites as Rebecka had, and one more in the eastern tip of the island. So some pictures. When we go regional, it means we have to go to the field. We have to take lots of samples from colonies in these amazing subantarctic islands. And yeah. So yeah, we had nice times. So I told you we knew from this paper in 2016 from Butz and collaborators that they presumably used different areas during winter, these two groups. So they tagged individuals in Malvinas and in Isla de los Estados. You see the Malvinas area of use in violet and the Isla de los Estados in green. And you see that the birds from this north group, the forage in northern waters, and the southern forage in southern waters. So if you followed Rebecka now, you could tell me which signature of carbon isotopes we should have in each area. So we found this correlation between this foraging strategies, these differences in foraging strategies within the genetic analysis. And so we found this south cluster that was compounded of these colonies, the colonies from the south, from southern Chile, and from Isla de los Estados, the two colonies grouped together. And then we found this north cluster that got together the Islas Malvinas colonies, the three colonies from Islas Malvinas that you can see here. All the individuals from Isla Pingüino were assigned to the same group, to the north colonies. So we found here that this colony was founded by Malvinas individuals. And we also found that the stranded individuals were also from Isla Malvinas, even the ones that are really close to the southern tip where the other cluster is breeding. So apparently these Malvinas individuals forage in all this area, in all the coastal Patagonia, and in recent times they founded this new colony. So about the carbon, the isotopes in carbon, what we found mainly is a south to north trend in the values. So here you can see the southern values for the values for the southern colonies, and here you can see the Malvinas and Isla Pingüino values. So you can see this trend that mainly coincides with this model that Rebecka showed you before. And you can see this, where these red colors are, where the less negative values appear. There are these values, and the same here, we get to these greenish, bluish values in the southern tip. So what we see here is that they are using different areas. As Rebecka told you, they are foraging in different areas. So during the pre-molt and the early molting period, they are using different areas. The same thing that happened with the satellite telemetry tags. So during winter and during pre-molt, they are using their foraging areas are segregated. Same thing here. What we did here was modeling this carbon and nitrogen values, and we tried to estimate the foraging niche, or the isotopic niche in this case. And we found the same thing. Again, we found this trend that the individuals from the south have these lower values again in nitrogen, and the same for the north. They have higher values for carbon and for nitrogen. So again, we see this south to north trend in both isotopes. So the nitrogen was not modeled. We don't have a model because it's quite hard to model nitrogen, but we see this same trend. So this could be explained by trophic position. So we did this evaluation. I will show you in a minute that. But I want to show you first this, that I found it like quite curious, is that the higher the the higher niche with the niche width, so the size of this ellipsis was higher for the Malvinas individuals. As you can see here, this is the standard ellipsis area. So you have higher values for Malvinas with lots of variation, but still higher than the ones from the southern group and the one from Isla Pingüino. So it's interesting to see that these birds that are doing some foundation of new colonies and are also dispersing around all these areas. I'll show you in the next map. Around all these areas are the ones that have the biggest, the widest niche. I know the more wide niche. So that's what we found for foraging. So now I'm gonna jump up into our mercury results. We found the same trend as Rebecka, but now we can see that the hotspot is quite localized. So we know that from, sorry, I jumped back a second, back here. So we know that these birds from Isla de los Estados and the southern tip of South America are foraging south during winter because of the satellite telemetry, and during pre-molt because of the carbon isotopes from the feathers. And we see that these birds are the ones that have the highest values that are quite similar to the ones that Rebecka found, both in both islands, in both colonies, sorry, from Isla de los Estados. And the ones from Malvinas, in spite of having this great variation in the size of the niche and this weather niche, they have this lower value. So presumably what we can assume is that the mercury values here in the shelf, in the Patagonia shelf, is quite lower than the mercury that is in the southern area near this is the subantarctic, sorry, I don't have the labels, but this is the subantarctic front that you can see in gray here. And here, the slightest line here is the polar front. So when these birds go down here and forage, they are bringing back the mercury from these areas. And what I promised, this is the trophic position, the same graph Rebecka showed you before, and the mercury concentration, but from this area, from the Patagonia area. So what we can see here is the trophic position, but this is the model output, so it has the variation from the model, and we have in the y-axis also the variation from the model output of the mercury. So what we can see is that the northern group, no matter what trophic position they have like really low values compared to the ones in the southern tip, which are the birds from Isla de los Estados, which have in this study, at least we found they have an intermediate value. They don't have these lower threes that Rebecka found. We found this 3.5. We did another approach, but probably there could be difference between years, so that could be the issue. But still, the thing is that it's completely independent of trophic position. So again, the same thing, and we are kind of localizing this mercury hotspot to the southern tip, but really the southernest tip of South America. And I'm going to show you now this work from Sami Dodino, who worked with Rebecka as well, and they worked like really regionally in this, like locally maybe, in this 3 colonies of Magellanic penguins. So they have this beautiful trend from inshore to offshore, and you can see the mercury pattern. So the mercury trend here is lowest in the inshore and highest in the offshore. So as you go out to the open ocean, you get more mercury. So again, we are getting the same information from different birds regionally. And I want to close up this presentation showing the implications we found, or we we discussed, and we're still discussing maybe for this study. So this is Isla de los Estados. Suppose this is a model of Isla de los Estados, and this is the part of the Rockhopper and Magellanic penguin here colonies. And what we see is that these birds, these penguins, go out and have some fun in the ocean and travel long distances, even up to 5,000 kilometers during the winter, and during the pre-molt, almost 300 to 700 kilometers from the colony. So they are traveling great distances, and they are bringing from the food they find in these areas, they are bringing nutrients, of course, but at the same time they are bringing mercury to the colonies. And how do they bring these nutrients? Of course, mainly the nitrogen they are getting through their feces. So they poop there in the colony, and they throw nitrogen to the colony and to the island, and that's an input of nutrients for the plants there for sure, and for the trophic the food web, in the terrestrial food web in the island. But they're also bringing mercury. And how they're bringing this mercury? Because of the feathers. So they are molting in the colonies during February to March, and they are depositing there all this mercury. And at the same time, what we found in a recently published paper by Ulises Balsa, we found that these nutrients and mercury is also transferred through the food chain. So this striated caracaras that Uli is working with, it's a bird of prey that they prey on chicks. So they deserve it. They get the mercury from the chicks. And they are incredibly, we found the highest mercury in any South American bird feathers. So this is quite like really important. So we are seeing like these penguins having this huge amount of mercury, but at the same time it has lots of consequences in the terrestrial environment. But we can see it in the biodiversity from the island that depend on these penguins and forage on these penguins, that they're having these mercury values that are up to 70 micrograms per gram. So it's like quite a high value. Remember, these are having like, I think the highest I found is like 7 to 10, something like that. So this is quite high. So we can use these ecological tracers, the carbon, the nitrogen, the mercury, as tracers for trying to trace, for trying to measure the relevance of the flux of nutrients and of the relevance of this input that comes from the seabirds. So all this energy and all these nutrients and all the pollution that comes from the ocean is mainly driven by these birds. But we could do what we haven't done it yet, but we could measure with these tracers the relevance of this. Is this, I think I have it in the next one. Is this input of mercury and nutrients relevant for the terrestrial environment? So would we see a trend from the colony to the inland of this island of mercury concentration? So highest mercury here and a trend going lower and lower as we move in the island, or not? Or is it just a local effect and it's just affected here? Do these birds have consequences? Are these birds in danger because of this higher values of mercuries? And at the same time, what we could be analyzing, and this is related to my genetic analysis that I have done, but I haven't started yet. But we could try and see if we are seeing like a new selective pressure. Maybe not new, maybe this is over hundreds of thousands or maybe millions of years there, but is there a selective pressure here? So are the birds from Isla de los Estados, state and island in English, from Isla de los Estados more resistant? Do they have a resistance to mercury? Do they have a way of excreting? Do they have a mechanism, a better mechanism of mercury excretion? Do these birds from the terrestrial birds, the birds of prey from Isla de los Estados, have a better way of excreting mercury? And we could compare this to the Malvinas birds that have like lower, or even to Isla Pingüino birds that have lower values of mercury. So is this happening or not? So that's our questions that are coming, and as now I'm open to your questions. These were mine.
Thank you very much. If you want, you can stop sharing your screen.
Okay.
Nico. And so what we are going to do now is you can, if you want, you can turn on your cameras. And if you have questions, you can type them on the chat box, or you can lift your hand and we will call you. And if you're on YouTube, we have people monitoring the YouTube channel, so we can also read your questions here and answer them.
Great. Sorry, I wanted to show this acknowledgements for the whole thing that we did. These are all the people that collaborated with us and worked with us during this time. So yeah, thank you.
Great. So I think I will start with one question, and I'm just wondering, because you have these two populations with differences in genetic structure and foraging ecology, are there any environmental barriers that are separating those two? Because they're quite close, actually. The few that you found in Tierra del Fuego that are not part of the Staten Island or Isla de los Estados, that are still part of the population of Islas Malvinas, are still different. So are there any environmental barriers that could explain the differences that you see?
I can take that one. So the main no, the quick answer is no. The water that comes from the southern tip of South America, that comes through all the circumpolar current, so those water masses are quite homogeneous. So and the water comes from south to north, and mainly at these distances, I mean and because the difference of temperature, salinity is like so low for these penguins. I mean it's no barrier for them. Maybe if we are seeing, I don't know, the Northern Rockhopper that I showed you, that they are like 5,000 kilometers from one colony to the other, they have to travel like long distances. Maybe you could think of differences in the waters and in the current. So, well, yeah, there could be. But here at this area, I'm pretty sure there are no barriers for this bird. So yeah, the main explanation we had for this segregation is foraging area. So they're foraging in different areas. They have different like selective pressures in these two areas, and maybe mercury is one of those. Maybe phenology. So the summer or the food comes first to Malvinas and then goes south like to the south. So maybe that's another reason that could be driving this divergence. But for sure, environmental would be like difficult to find something.
Yeah, I think I can add that whatever the environmental aspect is going to be something very, just add to what Nico said, it's going to be something very subtle. And I think that that's a great hypothesis about the timing of the transition of food, of moving. If food is moving from south to north, you could be talking about time. You could be talking about differences in temperature that and between when it goes from one place to the next. But it's got to be something really, really minor when it comes to those environmental conditions. And we just don't have, as far as I'm aware, we don't have any measures of that in those marine systems. So finding data on those subtle oceanographic differences in the water as far as we have looked high and low and rabbit holed our way through it all, we haven't been able to find data that allow us to actually test those hypotheses.
Thank you. Tim.
Yeah, thanks. And thanks for a wonderful AFO Café. I learned a lot. And part of my ignorance of this system was that I wasn't thinking about striated caracaras for potential bioaccumulation. So that was interesting to me. The things that came immediately to mind would have been like sheath bills and giant petrels. So are these sites a little bit too far north of where those birds are, or are there other potential predators active in these penguin colonies that could be bioaccumulating as well?
So yeah, for sure. We have gulls, we have petrels as well. The thing is that the caracaras are easier to capture. So yeah, and Uli was doing his PhD on caracaras. So yeah, it was like this opportunistic.
So the others are there, but you had the ability to work with the caracaras.
We don't know, but I would suspect that they have like higher values like petrels, and yeah, for sure they should have higher values if they are foraging there. But the thing is, like, we should know if they're actually foraging there. The ones that breed there, maybe they're foraging like quite far, but if they're foraging south, I would guess that they should have higher values.
Yeah, in general, where you find, like, so in the Antarctic, where you find petrels overlapping with the penguins, they do tend to have higher mercury levels, and that does tend to be associated with trophic position, because a lot of them are just like those caracaras. They're eating the juveniles, they're eating the eggs, things like that. But you also do run into the issue of the really wide foraging area for petrels. But a lot of people have done work on those types of seabirds. Sheathbills are always such an interesting one, because what they're foraging, like what their niche is anywhere from like penguin poop to more penguin poop or krill. And so I am not aware of papers looking at them. I think that, like Nico was saying, it comes down to access on these islands. Like if you're on this island full of birds, picking something that's like in the thousands that you can walk up to, or we got fortunate that Uli was doing his work on those caracaras that, again, they were already catching, tends to lead to those type of things.
Yeah, I would add that Sami, our colleague that she's working with Magellanic penguins, she found also difference between juveniles and adults. So mercury seems to be, tends to be accumulated through life. So adults having more concentration. So I was thinking of which would be the easiest way to sample these petrels would be to go to the colonies and get some chicks, but you would be getting like just a partial view of this thing. Maybe lots of input from the parent that is feeding at that time. So yeah, that's maybe the main like complications. Like catching the adults would be harder.
Thanks for that.
Do we have any questions on YouTube? I don't know if Matt is monitoring or somebody is.
No questions on YouTube.
Okay.
But a couple nice comments. They enjoyed the talk.
So I'm going to ask another question, and I have to admit I'm really ignorant when it comes to mercury and mercury levels and what they do. And so what's the effect of mercury poisoning on birds?
Yeah.
And what, like, there are some birds that are endangered, vulnerable. So what should we be looking for in those birds?
Yeah, that's a great question. It's something that I definitely that I mentioned but didn't touch on specifically. And so for birds in general, reproduction is the most sensitive endpoint of mercury toxicity, and that even very, very low mercury concentrations have been linked to decreased hatching success of the egg, decreased chick survival. It can affect clutch sizes. That tends to be something that we see more often in birds that have larger clutches, whereas these penguins are having one or two. 1 or 2 very large eggs. But at these levels, we would be looking at those reproductive within a season, number of eggs you lay, the proportion that hatch, the survival of those offspring from hatch to fledge is another big one. And so one of the things with the rockhoppers that was cause for concern with the southern populations was the population decline that they're experiencing. And so it was because their levels were sort of above that adverse effects levels where you would expect to start to see decreases in reproductive success. It was curious to know if that could be contributing. Now, this is not something that we studied. I would love for somebody to pick that up and do that. But yeah, because it does accumulate over the course of the life, you could look at things like lower productivity over the course of their life, so fewer reproductive events. There's plenty of, at very high concentrations, it is a neurotoxin. So there have been plenty of studies that look at the effect of mercury on migration, returning to phylopatric nesting sites, things like that. But mostly we're looking at subtle effects on reproduction. Immune system, that's another big one that's come up a lot recently on immunocompetence. And so mercury has been shown to decrease immunocompetence in the adults in particular that have been studied. And so again, that could affect survival and the ability to take care of these really hard to raise offspring.
Yeah, what we found in the caracaras that was really interesting that we published it yesterday. I'm going to share the screen with the figure from the paper, if you can see it now. Yeah. Mm-hmm. So this is the one with that I told you that had the highest value. It's 85. It was not 70. So what we saw is the one of the legs. So they had it like stiff. They couldn't move it, and they had this overgrown claws here, and this could be maybe explained because so mercury deposits in these keratin tissues like feathers and nails, and maybe the toes are not working because of that. Of course, we didn't test this, but we found like it was curious to find the highest value with this leg that could not move at all, and it was an adult.
And if you think about that difference in mercury concentration, so those penguins, those adult rockhoppers were coming in at 5 to 7 parts per million, and that caracara had 85. Yeah. And that's an animal that, I mean, those feathers are molted annually. So they're dumping. I mean, it accumulates through life and can be representative of that. But if you're eating penguins, that is a high mercury way to go. And we haven't ever seen anything like that in the literature before. So it was really interesting.
Yeah, and I would add that the penguins have this kind of collapse that they throw all the moltings, like I say,
catastrophic molt.
Catastrophic molting. So they mold all the feathers at the same time, and they release all the mercury there. So we are seeing like the highest possible value of mercury for the rockhoppers, and in the case of the caracaras, that's not so, because they are molting like during the summer for sure, but in a longer time. So we are seeing like this well, I don't know how to say it, but this like releasing it in a longer time.
Release.
Yeah, slow rate of release. So this is even worse for these birds. Yeah.
Any other questions? I don't see anything on the chat. I don't know if there are people that have other questions. No. And no more questions on YouTube? Okay, nothing on YouTube. Okay, well, no. Well, thank you so much. This was really amazing and very clear. Thanks a lot. And congratulations, Nico, on your PhD.
Thank you.
Okay, so yeah, there we go.